You heard, yeah. Everyone is here who is necessary for the poster session. Uh, we have the poster session now during the break in order to get in time a little bit. It was planned like this. So we changed a little bit the program because the first one is not at the moment on site. And so I asked the number poster 6, prevention of public health harms by switching from tobacco smoking to alternative nicotine products. Alexei Alexandrov.
Hello, I'm Alexei Alexandrov from Belarus. I'd
Wonderful.
like to present results of studies conducted in Belarus which focus on prevention of tobacco-related health harms by switching to alternative nicotine products. Economic analysis was conducted by Dmitry Ruzanov and Vital Pisarek. Belarus population was decreased up to 50,000 during 5 years, mostly because of excessive mortality of adult males. One of the reasons of this is smoking-related illnesses and deaths. According to Belarus STEPS surveys, about 50% of adult males in 2016 were current smokers. There was slight decrease in smoking in general population during the last 5 years. One of the possible explanations of such decrease is spreading of the use in 2020 heated tobacco products to 3% of population and electronic cigarette to 6%. In 2021, result of sociological study conducted by Institute of Sociology and toxicological one by Belarus State Institute of Metrology dedicated use of HTP were published. Sociological study involves 1,500 respondents. Both electronic cigarettes and heated tobacco product consumers reported about decrease of smoking intensity. Almost half of heated tobacco product users quit tobacco smoking and perceives this product as less harmful. Toxicological study did comparison of heated tobacco product aerosol and tobacco smoke contents. It showed that first one reduced exposure to different toxic and cancerogenic substances up to 1,500 times. Economic analysis used disability-adjusted life years as assessment tool of smoking-related loss of years. In the Belarus in 2019, every fourth year lost from smoking as risk factor, and 1 in 6 years lost from smoking-related causes of death. The economic benefits of implementation of tobacco harm reduction in Belarus were calculated under various models in different levels of prevented harm. First model shows decrease of awful mortality up to 9% with saving of $4 million. Switching of 200,000 smokers to heated tobacco product could save 5% of daily lost and about $30 to $70 million saving per year in all genders. Third model, find loss saving are from 5,000 dailies and from almost $20 to $40 million. Last economic model based on assumption that every 5 years, 2.3% of tobacco smokers switch to heated tobacco product. Possible preventable harm during 10 years will almost 300,000 years of daily and saving at $2.5 billion. There are some conclusions on this side, but I'd like to finish my report with words of Mahatma Gandhi. Be the change you want to see in the world. Thanks for your attention.
Thank you very much. Very interesting data from Eastern Europe. Maybe calculations may be the same in some other parts of the of European countries. Any comments? Okay. Thank you very much for this really interesting input, and keep on searching about these topics. I believe it's very important for the economics. Thank you.
Thank you for opportunity.
Next poster is new realities in use of different tobacco delivery devices. Egul Zanatulina from Russian Federation. I ask if she is present on.
Good afternoon, dear participants and guests of the conference. My name is Eguzina Turina, and I want to present to your attention a report on the topic New Realities in the Use of Different Tobacco Delivery Devices. When studying the literature data in recent years, attention is drawn to the growing interest in lung lesion associated with the use of electronic cigarettes. There has been an increase in the use of vapes and electronic cigarettes instead of traditional cigarettes, and an increase in the number of middle and high school students smoking electronic cigarettes compared to adults. The aim of our study was to identify differences in groups of smokers using different tobacco delivery devices. To conduct the study, we used a questionnaire on the use of various means of tobacco delivery, the Horn questionnaire, the Fagerström test, and the scale for assessing the degree of readiness to quit smoking. 57 smokers were interviewed and divided into three groups. The first, those using regular cigarettes, the second, electronic cigarettes or vapes, and the third, users of heated tobacco product. The mean age of the groups overage 40, 39, and 35 years respectively. As a result of the study, it was found that the strongest nicotine addiction is in people who use heated tobacco products. At the same time, they have the highest motivation to quit smoking. In the group of vaping smokers, again the background of low nicotine dependence, low motivation to quit smoking is noted. This may be due to low awareness of the dangers of vaping among the population and especially young people. When studying types of smoking behavior, we found that the support type prevails in all groups. This type is associated with situations of excitement and emotional stress. The first type dominated in the heated tobacco product users group and was less common in the vapers group than in the others, which confirms the level of nicotine addiction in this group. And the relaxation type was more common in the group of vapers than in other groups. Such smokers usually smoke only in comfortable conditions. Such types of smoking behavior as a reflex, gain, and stimulation were rare in all groups, so we didn't include them in our presentation. We noticed also that 84% of regular cigarette users combined smoking with alcohol. Almost 19% of persons from the heated tobacco product group combined smoking with drinking coffee. Conclusions. The severity of nicotine addiction was greatest in the group of people using heated tobacco product, and the lowest motivation to quit smoking was in the group of vapers. In all groups, support was the predominant type of smoking behavior. Relaxation was the prevalence in the vapers group, increased in the group of heated tobacco product users. Most smokers, more than 70%, tend to combine smoking with alcohol or coffee. So we think that the identified differences may be useful in developing individual smoking cessation programs. Thank you for your attention.
Thank you very much. I believe it was an interesting input coming also from the Eastern Europe. Is there any comment in the audience? When there is no one, we continue to with poster number 8. heat not burn tobacco products and their relevance in oral health. This is a video.
I would like to present a mini systemic review of heat-not-burn tobacco and their relevance in oral health. Most people are unaware of smoking effect on their mouth. The chemicals in cigarette smoke can linger in the mouth and lungs for hours, causing the characteristic smoker's breath. The nicotine and tar in tobacco can make teeth go yellow in a very short time. Smoking can increase the number of bacteria in the mouth that cause gum disease. 2/3 of mouth cancer cases are linked to smoking. The dental team can play an effective role in finding the pathologic problem in the mouth and in counseling their patients to quit. Present studies suggest that HNB tobacco products have the potential to be a reduced risk product for public health compared to traditional cigarettes, especially when indirectly considering the potential effects on the chronic disease, which are traditionally linked to cigarette use. To do the research for this systematic review, the PubMed ISI Web of Knowledge Scopus databases were searched up to August 2023. The keywords used were heated tobacco products and oral health. This systematic review intends to answer the question, are tobacco heated systems less harmful to oral health when compared to conventional smoking? There was evidence from 11 studies that tobacco exposure caused staining of dental hard tissues and dental materials. It can be concluded that evidence support the clinical observation e-cigarettes and HTPs cause staining to a lower extent than with cigarette smoke. For gum disease, results from all studies show that culture exposed to HNB system aerosols exhibited less cytotoxicity and minor morphological alterations, thus supporting the concept of less harmful tobacco products. Similar observations were made regarding exposure effects on human lung epithelial cells. The research conducted cancer was all in cell-based, and we cannot find any one about oral cancer. Experimental studies are needed to assess if the relatively lower dosage of toxin released by the HNB is capable of inducing malignant transformation in oral epithelial cell line and mouse model. One randomized control clinical trial was reported. Results indicate that routine clinical treatment improved the course of periodontitis similarly in cigarette smokers and HNB users. The beneficial effects of this treatment might mask the changes between the groups. The health risks of heat-not-burn cigarettes are unclear. Much of the research has been undertaken by the tobacco companies and are laboratory-based, as the products have not been available long enough for us to fully understand their effect on oral health. More long-term research is needed.
Any comments? I believe it shows the differences between the classical snus, the pouches, and other heated tobacco products. Very interesting in the combination or in the differentiation of the possible problem of a local effect. Any comment? Any discussion for this? So then we do the next one is the number 9, analysis of awareness and readiness of patients with malignant neoplasms to quit smoking as a factor influencing overall survival, from Sofia Safina, Russian Federation. So please.
Greetings, dear colleagues. I'm Aygul Fayrushina, doctor of the Safina team of the Professor Sigal Republican Oncology Dispensary. Let me present the result of our research analysis of awareness and readiness of patients with malignant neoplasms to quit smoking as a factor influencing overall survival. Objective of our study is to analyze of awareness and willingness of patients with malignant neoplasms to quit smoking as a factor affecting overall survival. According to this slide, the larger percentage of smokers was found among men over the age of 50 years. The smoking experiments was more than 5 years in 82% of patients, and the manifestations of the habit was up to 80 years in 57.6%. Many studies show that willingness and motivations to quit tobacco increases when a patient is diagnosed with cancer. According to the results of our study, 88% were those who tried to quit smoking. However, only 40% succeeded in quitting smoking, and only 10% of respondents didn't smoke for more than a year. In diagram on the left, 67% of respondents smoke more than a pack of cigarettes a day, and 45% of patients have a history of concomitant pathologies from cardiovascular and respiratory system. 63% of respondents had high motivations to quit smoking. These were patients who made this decision on their own. Among them, 80% of respondents had more than 5 attempts. Our conclusion: the high compliance identified indicates the need to provide patients with tobacco cessation assistance. Smoking cessation should be a component of cancer treatment. Patients who are unavailable or unable to quit smoking can be advised on the tobacco harm reduction concept. The combinations of following factors will allow us to influence the problem effectively. There are introduction to clinical guidelines, component of cancer treatment, restriction of tobacco advertising, restriction to the sale of tobacco, school educational program, promotion of tobacco cessation, tobacco harm reduction, counseling the treatment of withdrawal syndrome. Thank you very much for attention.
Thank you very much. I believe it's an interesting thing because patients should have the possibility to change, but not the absolute need and the pressure in such a situation when they have a cancer. We have to give him a possibility, and this is a wonderful thing when harm-reduced products will be available for these patients. Any comments? When not, so we proceed to number 10, comparative toxicological assessment of cigarettes and new category products via in vitro multi-complex proteomics platform. This is presented by Nikos Tsolakis with further investigated by David Smart, please. So be happy to be here.
Yes, it looks like I should have pre-recorded it, doesn't it, really? I'm so glad that so many of you managed to turn up for my poster presentation. I believe Kalispera is the correct word at this time. Good afternoon. I'd like to thank you for inviting me here to speak today on the comparative toxicological assessment of cigarettes and new category products via in vitro multiplex proteomics platform. This is a collaboration, as our compare gave description of, between British American Tobacco, of which I'm a member, and Protavio in Athens and also Athens University. We've also heard mention of Public Health England a few times throughout this weekend. Well, it's not weekend now, isn't it? These couple of days. Started on a weekend for me. Which, anyway, Public Health England are a strong advocate of tobacco harm reduction, and as such have suggested that vaping may bring about as much as a 95% reduction in harm over other products. We chose to investigate this as part of the study and extend it to sort of other tobacco related products, so heat not burn. Oops. And as such, we took the three main categories that we've been discussing here over this conference of vapor and heated tobacco products, and using cigarette 1R6F as a control. So it was EPEN3 and the Glow standard device, using a Borg Volt LM4E as the source of puffing, using the Health Canada Intense for the cigarette, Health Canada Modified Intense, which is minus the taping of the vents for the Glow, and CRM 81, which is Caresta Method 1 for the vapor products. And this was used to puff through cell culture media held in a glass impinger. This was scaled across the three products, which gives an added level of the kind of differential between them. So we used 8 puffs for the cigarette, 40 puffs for the Glow product, or the heated tobacco product, and 200 puffs for the e-cigarette product. We used NCI H292 lung carcinoma cells. We initially serum starved these for 2 hours to relieve a baseline of phosphorylation before exposing them to the AQE samples at a series of dilutions for 2 hours. The cells are then washed with cell culture media, and this was again replaced for a 48-hour culture prior to harvest. For the phosphorylation signaling proteins, this was slightly modified in that the cells were harvested immediately following the 2-hour incubation. As you can see here, we did a standard heat map with the reds, the darkest red indicating the highest upregulation, when the darkest blue, the greatest downregulation of the proteins that we looked at, analysed, with the inflammatory proteins on the left and the phosphoproteins or signaling proteins on the right. You can see that the different categories actually segregated themselves nicely by hierarchical clustering, with in the brown, the 1R6F or cigarette being most distant from the vehicle control, with Glow and e-cigarette following on from 1R6F moving towards the vehicle control. For the inflammatory proteins, we saw some overlap between the vehicle control and the vapor product. Again, we've looked at the same data, this time by volcano plot, and you can see there's upregulation by, particularly in the 1R6F or cigarette products, of a number of proteins, in particular interleukin-1 alpha and beta, CREB1, CJUN, MEC1, and P38 MAP kinase. You can see that these are downregulated when you go to Glow, again, following along, as you saw in the hierarchical clustering. That only in Glow, the only protein that continues to be upregulated is the MEC1. And when you move to EPEN3 or the vapor product, you don't see any upregulation, bearing in mind obviously the differential we had in the puffing regimes of the 8, 40, 200 puffs. So in summary, our results are consistent with the risk continuum of moving from cigarette at one end through heated tobacco products through vapor towards cessation or non-smoking. This was a multiplex proteomics platform, and it proved to be a robust, reliable tool. You saw some agreement between none of the replicates, so there was 5 replicates in each of those treatments, and it may provide a suitable screening tool for reduced risk products going forward. And using inflammatory and signaling markers could help provide comparative risk assessment also going forward. Obligata.
Thank you very much for these interesting results. They fit, I believe, together with the other results we have. They are further puzzle to show where we stand with reduced products. Any comments, any question? Now you have a possibility, a real life possibility.
It certainly gives you an opportunity to put some mechanistic processes in place to explain the potential of risk reduction going forward. So it gives you a mechanistic story to kind of demonstrate those risk reductions.
Yeah.
Yes.
Just a quick question. Certainly, we back in that much deeper pockets, if you can go money to spend than a university. About with this kind of experiment, what does this kind of equipment cost? Excuse me, if you can go to the microphone so they can hear you. Dr. O'Leary from the University of Catania. Again, the tobacco corporations have deep pockets. They have money to spend, whereas we in academia are scraping for every last euro. I was just curious how much this kind of equipment costs to get a lab set up.
So we're actually going to see the collaborators tomorrow. So our input into this was fundamentally sending the extracts to the collaborators from this. So this was a university institution that effectively did the study. So I'm not sure what the costing is, but it was an industry university. And with regard to the deepness of the pockets, I wish you'd tell my boss that when I'm asking to buy some stuff.
You should, because at University of Catania we have the replica studies, and I remember it put a serious dent into our budget. Well, worth the cost, of course, of admission, but is this something that we're aware of, just like Dr. Livolti was asking me how much time and money?
Yeah.
We're always working with those.
We're always willing to work with academics, but so there's limitations on some academic universities are not allowed to work with tobacco industry, particularly in the UK, for example.
So hope we see these results fast in the peer review journal. Thank you very much.
Indeed. Bye.
So next presentation is number 11. We are connected. 11 it's no show. It's right. So we come to the number 12, quantitative adverse outcome pathways, a new prediction tool to assess human risk of decreased lung function when switching from combustible cigarettes to heated tobacco products. Please.
Good afternoon. I'm the last guy between you and lunch. So my name is Amin Shokrala. I'm from Philip Morris International. I'm replacing my colleague Carol, who couldn't join the meeting. And this poster is about using QAOPs or quantitative adverse outcome pathways as a predictive tool for hazard assessment in the context of tobacco harm reduction. Can we go to the next slide, please? Thank you. So for those of you who do not know the concept of AOPs, so AOPs, it's a conceptual framework that allows you to collect and organize mechanistic data into a causal sequence of events. So it often starts with a molecular initiating event that goes through a number of other events leading to an adverse outcome. So the AOP we are interested in today is start with a molecular initiating event, which is oxidative stress, that will lead to a decrease in cilia beat frequency. The relationship between events, it's called key event relationship, and this relationship in the QAOPs can be mathematically modeled to allow the prediction of the next key event. So the decrease of cilia beat frequency, or CBF, will lead to a decrease in mucociliary clearance. And this last event will lead to the adverse outcome, which is a reduction in lung function. So in theory, when you are constructing an AOP, you would like to have only human data. But for obvious ethical reasons, some of the data cannot be generated from humans, so you need to replace this by in vitro data. So in this case here, the colleagues, they combined data coming from in vitro, in silico modeling, and clinical data. So the oxidative stress was evaluated using gene expression data, and there is some artificial intelligence here to model the data and to predict oxidative stress. From the same organotypic cultures, the colleagues, they also measured the CBF, the cilia beat frequency, using high-speed digital video recording. So once you have these two data, so you can model them to try to predict the CBF decrease as a function of gene expression data, which is informing you about the oxidative stress. So this is what you see in this first graph here. This is a modeling of the first key event relationship. So what you see in the x-axis is the difference between 3 different exposures that are cigarette smoke, HTP aerosol, and air control. It's a bit a complex graph, but the most important information, this is the difference. So when you are close to 0, meaning there is a little difference between the exposure and the air control. And this is what you see in the purple dots are the ones are the values from HCP aerosols, and you see they are close to 0, meaning that the cilia beat frequency decrease is not affected by the exposure, but also the gene expression values informing about the oxidative stress. So the next step is to predict or to make a relationship between the decrease in cilia beat frequency and the mucociliary clearance. So the small squares you see in the second graph here are generated using fluid dynamics. And so in the x-axis you have the CBF, this is a ratio, meaning if this is a ratio of the exposure versus the air control. So if it's close to 1, meaning there's a little difference between the exposure and the control. So what you see here is that the HTPs are very close to 1, meaning there is a little impact of the exposure on CBF. And what you see in the y-axis, this is the predicted MCC using fluid dynamics. And here again, you said there is little deviation from 1. The box plots are human data coming from literature. So the authors here, they measured MCC using the saccharin transit time test in non-smokers, cigarette smokers, former smokers, and HTP switchers. And you could see that while non-smokers and former smokers and also HTP users, the box plots are close to 1, there is a reduction in MCC in the smokers, meaning there is a recovery potential when people they move from smoking cigarettes to HTPs or quitting completely smoking. So the last graph is the mathematical modeling of the last key event relationship going from the reduction in MCC to a reduction in lung function as measured by spirometry FEV1. So here again, here the baseline or the reference is former smokers. You see this is 0. And the cigarette smokers, you see there is a reduction in the MCC and predicting also a reduction in FEV1. For HTP users, there is no data, though this was modeled using the MCC data. And you see that based on this modeling, the HTP, the values from HTP are somehow positioned close to former smoker than to cigarette smoker. So if you combine all this mathematical modeling, the risk of having the adverse outcome, which is a reduction in lung function, if someone continues smoking, it's set to 100%. For a former smoker, it's set to 0. And if we combine all this data, this modeling is telling us that for HTP switchers, the reduction would be about 80%. But there are no real data outside, it's only predictions. So as a conclusion, so to construct this kind of IOPs, you may need in vitro, in silico, and clinical data, and this can inform you about an adverse outcome, which is lung function, that sometimes you cannot really measure in a real patient. Yeah, that's it. Thank you. If there are any questions.
Thank you very much. I believe it's maybe a link to the exacerbation rate, maybe reduced exacerbation rate, what we have seen in users of heated tobacco products. A possible explanation specifically due to the fact that this is one airway from the nose until to the lung. Further questions, comments? You have the possibility. No one? Thank you very much.
Thank you.
So we ask if we have still someone in the line. No one? This is number 12. Here we finish. Is it right? We have one? So we finished. Thank you very much for your attendance, and try to get some bread or some drinks still on until we start with the next session. Thank you very much.